Okay, everyone, so now let's take just a brief look at the eukaryotic cell cycle. So what is the cell cycle? So when I talk about the cell cycle, I'm referring to the process that allows one parent cell to become two identical daughter cells. I don't know why these are called daughter cells and not son cells. But sometimes they're called daughter cells. And so identical cells. So, and they are genetically identical too because mitosis is part of the cell cycle. So the nuclei in these cells should be genetically identical. And traditionally, the cell cycle is divided into four main stages. We have S phase, G2, M, and G1. And then sometimes there's another phase over here called G0. So now, how does this cell cycle work? Okay, so the cell cycle describes the various phases the cell will go through when it's dividing. And this G0 is a phase that, the cell is thought to enter when it's no longer dividing and making more cells. So G1, we're going to call these the growth phases. So G1 and G2, the growth phases. So the cell is preparing for something. In G1, if the cell is not entering into G0, the cell is preparing for S phase. And S phase, this is the synthesis phase. This is when DNA is replicated. So DNA synthesis happens in S phase, which is, kind of confusing for a lot of students because we talk a lot about what DNA does during mitosis. So how the cell, puts half of the DNA into one nucleus and half into the other during mitosis. But it's important to remember that the DNA is not replicated in mitosis. It's replicated way up here in S phase. So now G2, we can consider G2 as the growth phase where the cell is preparing for mitosis, and mitosis is going to be down here. Now, all of this up here, all of these phases that are not part of mitosis, we can group them together into a stage that is generically referred to as interphase. So they are all part of interphase. So if the cell is not in mitosis or meiosis, it is in interphase. Okay, so one thing I wanted to point out about the various phases is what the chromosomes look like. So we already talked about how during how the chromosomes condense during mitosis, and when they're not in mitosis, they're generally decondensed. So up here in these phases up here, G1, S, and G2, these are all part of interphase, so we would expect that the DNA of each chromosome would be decondensed. It would be very difficult to see under a microscope. But as I alluded to previously, I will diagram, even when I'm talking about interphase, I will diagram chromosomes in the condensed form. So during G1, we would expect chromosomes to look or to consist of a single DNA molecule. And this right here, this is a chromosome with a single DNA molecule, and it has a single centromere, and it has the chromosome arms, and that is, that's what we would typically, I would typically diagram a chromosome. So I'm going to label this one chromosome. Now, when the DNA is replicated, well, that single DNA molecule becomes two DNA molecules. So in G2, I'm going to represent the chromosomes like this. So this is the same chromosome, and by convention, it's also considered to be a single chromosome. So this is one chromosome. But there are two chromatids in this chromosome. Chromatids. Two chromatids. And these two chromatids, should have an identical DNA sequence because they resulted from replication of one DNA molecule. These are called sister chromatids. Now, assuming no mutations happen during DNA replication, those sister chromatids should be genetically identical. And they, by convention, we say they share one centromere. So the two sister chromatids share one centromere. And by convention, that's how we define chromosome numbers. We look at a cell under a microscope and we count the number of centromeres. So over here, we see one centromere. So that's one chromosome. Over here, we see one centromere. So this is one chromosome. These are both diagrams of single chromosomes. Now, to be complete, I could say this chromosome right here is made up of one chromatid. Okay, so now that we know what chromatids are and sister chromatids, and what the diagram of a chromosome that has already passed through S phase and now is in G2 should look like, we can talk a little bit about human chromosomes. So how many different human chromosomes are there? So I'm going to say there are 24 different chromosomes. So 24 different chromosomes. And some of you might be thinking, wait, humans have 46 chromosomes, right? So I'm going to say there are 24 different chromosomes, 22 maybe I should say different types of chromosomes. So 22 autosomes and 2 sex-determining chromosomes, sometimes just called sex chromosomes. So the 2 sex chromosomes are named X and Y. And the autosomes, so an autosome is any chromosome that is not a sex chromosome, any chromosome that is not a sex-determining chromosome. The 22 autosomes in humans are named, by numbers. So they're 1 through 22. And their numbers, for the most part in humans, the chromosomes are numbered by length. So with this information, we can predict that chromosome number 1 in humans is probably the longest chromosome. Number 1 is the longest, and we can guess that number 22 is the shortest. I don't think that's accurate. These numbers were based on before DNA sequencing was available, and they were based by looking at chromosomes under a microscope, and I think it turns out number 21 is the shortest. But for the most part, just knowing that chromosomes are usually numbered according to their length, with the lowest number being the longest. Knowing that is important, and you can probably guess which chromosomes are longer based on the number of the chromosome. So I have a diagram down here of the 24 different human chromosomes. Let's see, where is it? I had one. Oh, here it is. Okay, let's zoom out a bit. So here are the 24 chromosomes in humans. Now, chromosome number 1 and number 3 are considered to be the metacentric chromosomes in humans. There are no telocentric, and all the remaining chromosomes are acrocentric chromosomes in humans. So okay, so 24 different chromosomes in humans, but humans, like we have 46 chromosomes, right? So, okay, how does that happen? So, in humans, right, we reproduce when we reproduce naturally, and we might not always need to do this. For fans of The Expanse on Amazon Prime, what do we have? Or the book series, The Expanse book series. There is a character, James Holden. He's like the main character of the series. He is the result of, he has 12 parents. He has 12 parents, so essentially DNA from 12 different parents combined to make James Holden, obviously using some sort of in vitro fertilization and genetic engineering. But for now, most humans are a result of what? Fertilization of an egg cell by a sperm cell, or technically a secondary oocyte. We'll talk more about that later. So human sperm have 23 chromosomes. Human eggs, 23 chromosomes. And these are considered gametes, right? Sperm and eggs. Technically, they are called gametes. And of those 23 chromosomes, 22 are autosomes, and 1 is a sex chromosome. For sperm, it's either 1X or 1Y. And for eggs, 22 autosomes, so 1 copy of each of the human autosomes, plus 1X. So you can see well, I'll tell you that the sperm and the egg each have 1 set of human chromosomes. So a set of human chromosomes includes 1 copy of each autosome plus 1 sex chromosome. That makes 1 set. So there's 1 set of chromosomes in the sperm, 1 set in the egg, and they come together to make the zygote. Now the zygote is the first cell that results from fertilization of the egg by the sperm. And the zygote has 46 chromosomes. This is humans, this isn't all animals. So 44 autosomes and 2 sex chromosomes, normally. And what? 2 sets. So the zygote contains 2 sets of human chromosomes. And this is where we come into this concept of ploidy, or ploidy levels. So ploidy refers to the number of sets. So most human cells, starting at the zygote stage, are diploid. So diploid cells have 2 sets of chromosomes. What types of human cells aren't diploid? So our gametes are haploid. So haploid means only 1 set of chromosomes. Our gametes are haploid. haploid cells have one set of chromosomes. Some organisms, most of their cells, like fungi for instance, most fungi are haploid. Most of their cells have a single set of chromosomes. There are many plants that are tetraploid. They have 4 sets of chromosomes, or hexaploid, or octaploid. Many of the fruits we eat are triploid, and these fruit are actually sterile. And we'll talk why a triploid organism, or a triploid offspring, I should say technically, is sterile. We'll talk more about that later. But this is what ploidy means. So humans are diploid. Most of our cells are diploid except for our gametes. They have 2 sets of chromosomes. One set inherited from our fathers and one set from our mothers. So if we were to look in a random human cell at a nucleus, not a gamete, so any other cell, and we were to identify, let's say, chromosome 1. I'm going to say chromosome 1 from the biological father. I'm going to indicate that with that little, what is this? This is the generic symbol for male, and this is the generic symbol for female. So, and let's say that's chromosome 1, and let's say this one over here, this is chromosome 10. And this diagram is not to scale. I put an X, the Roman numeral. I can't do that, right? Because our sex chromosomes are called X. So chromosome 10. Now, in that cell, because we know humans are diploid, and they have 2 sets, diploid cells have 2 sets of chromosomes, we should be able to identify another chromosome number 1 in here, chromosome 1 from the mother. The same thing goes for chromosome number 10. There should be another chromosome 10 in here. And these chromosome number 1s, these are homologous chromosomes. And so homologous chromosomes are chromosomes that have the same structure and gene content. Now, we can't say they have the same DNA sequence. And the reason we can't is kind of complicated. It's a little too early to explain why, but I'm going to try to anyways. So if that's a chromosome, and this is a homologous chromosome, and let's say this is a chromosome from mom, this is the homolog from dad. I should say homologous chromosomes are sometimes just called homologs, a pair of homologs. So let's say over here there's a gene for hair color, and let's say that gene has a certain sequence for making, I don't know, dark brown hair. And we're going to call that gene big A. The sequence of that gene, we're going to refer to that as big A. And over here there might be a gene for hair color. The one from dad has a slightly different DNA sequence. It's the same gene, but it's a different version of the gene. The sequence is a little different. We're going to call that one little a. Let's say that's for like blonde hair color or something. So the same gene is found in the same place on the chromosome, but the sequences are slightly different. That doesn't make the chromosomes non-homologous. These are still homologous chromosomes. Autosome number 1 from mom, autosome number 1 from dad. They have the same structure, they have the same gene content, the genes are found in the same order, but their DNA sequences may not be identical. Okay, so this is a pair of homologous chromosomes, and I hope you understand what the concept of homologous chromosomes is now. Okay, so now we are ready to talk about mitosis.